My name is Aoife Ni Mhaille and I have just completed my PhD with the Multiple Sclerosis and Stroke Research Group directed by Dr. Una Fitzgerald at the NCBS in UI Galway. The aim of my project was to determine whether proteins associated with the endoplasmic reticulum signalling pathway were present at raised levels in MS tissue. MS is an autoimmune degenerative disorder that affects the brain, the spinal cord and the optic nerves. MS is characterised by the loss of a fatty substance called myelin, which insulates the nerve axons. See here how the brain slice on the left contains multiple dark peach patches, which are absent in the normal non-MS brain. These patches are called lesions or plaques, and they are a typical feature of MS pathology. The tissue surrounding the lesion is paler because it contains more myelin. Myelin is produced by brain cells called oligodendrocytes, and each oligodendrocyte can myelinate a dozen axons. Within mammalian cells, a subcellular compartment referred to as the endoplasmic reticulum is responsible for the correct folding of secretory and membrane brown proteins. Sometimes the folding machinery within the ER isn't functioning properly or becomes overloaded with misfolded proteins, and this activates a signaling pathway called endoplasmic reticulum stress. The reason we became interested in the ER stress pathway is based on reports which showed that it was associated with cell death following stroke. Further reports indicate that oligodendrocytes resembling those seen following a stroke are present in MS tissue. So we put two and two together and asked the question whether these cells could be dying in an ER stress dependent manner. To answer this question, I analysed post-mortem brain tissue from the Belfast Brain Bank, the MS Brain Bank and Beaumont Hospital. This slide summarises how brain tissue donated by MS patients and normal volunteers is processed through the UK MS Tissue Bank. A special blue dye called Luxol Fast Blue specifically stains myelin, so when it is applied to MS tissue, it is very easy to pinpoint the location of a lesion, as shown in the next image. Here we can see the areas in the diseased MS brain which are white instead of blue. This means that myelin has been lost here as the lesion has developed. We placed thin sections of brain tissue on glass slides and applied a panel of antibodies to test our hypothesis. That means that we wanted to show that raised levels of ER stress proteins were present at the edge of MS lesions. This picture is an example of the kind of tissue staining we obtained. See how in the left-hand panel a dark brown staining is present along the edge of the MS lesion. This shows that a protein called CHOP is present at raised levels in the diseased tissue. When we move away from the lesion, the brown staining becomes less intense. Messenger RNA is the template used to synthesize proteins. Each protein has its own mRNA. Therefore, we next looked at the levels of the ER stress proteins via their mRNA template. First, we cut frozen sections using a cryostat. To make sure that the tissue and proteins didn't degrade, this had to be done once the machine was cooled to -15°C. After isolating RNA from the frozen tissue sections, we used a bioanalyzer to check the quality of the RNA. This machine separates the different molecules of RNA according to their size by forcing them to travel through molecular sieve or gel. A dye is also applied to the RNA which makes the RNA detectable when a laser light is shone through the gel. This is then translated into a black and white striped image, as shown for the different samples in the different lanes here. Each stripe or band corresponds to a different RNA molecule. The bands at the top of the image are larger than those at the bottom. In high quality RNA, the bands are clustered, so that some bands appear more intense or darker than others. Low quality RNA generates a more even kind of pattern, especially in the lower part of the picture. The RNA integrity or RIN numbers are shown at the bottom of the picture. Once we were happy that the RNA was of high quality, we used a method referred to as real-time PCR to back up the data we found at immunohistochemical levels. While carrying out this work, I was supported by NUIG College of Science Fellowship, the Foundation Office at NUIG and MS Ireland.